Hello, everyone. The webinar will begin momentarily. Hi, everyone. Linda Hyman here from the MBL, and I am absolutely delighted to welcome you to our last in a series of SES Lunch and Learns. Before I turn it over to Ken Foreman, who will be serving as the moderator for today's session, I just wanted to take a moment to thank everybody who participated in this series of events. They've been terrific. And a big shout out goes to both the faculty and our alumni, as well as the folks online who've joined us for these events. So really appreciate your being here with us. Also, today is a banner day in education in that our SES application for the 2021 season has gone live. And so we hope we'll be seeing lots of new faces here in the fall. It's been a crazy year, and we wish you all good health and good cheer, and take care. So with that, I'm going to turn it over to Dr. Foreman to lead us through today's exciting session. Thanks.
Well, thank you, Linda, and thank you all who are in attendance today. I want to remind you that as audience members, you are muted and your video is turned off, but we'd like this to be a conversation. So at any time, if you have a question, you can submit that through the Q&A button at the bottom of your screen. And at the end of the presentations, we will address those questions. Today, you will hear about the possible effects of changing climate and nutrients on the carbon balance of both temperate forest and Arctic tundra ecosystems. As we've emphasized in each of these talks, the MBL has a long tradition of training students from around the world by linking research with education. And concern about climate change and its effects on the biota is a theme that we have emphasized in the SES program throughout the 22 years that we've offered it. And during that time, SES has helped train nearly 400 students from more than 60 colleges and universities around the nation. Students in the program experience life as a scientist. They spend 20 hours a week in the lab and in the field, and they cap the semester by designing and completing independent research on questions they wish to answer. Most work on ecosystem is done on ecosystems here on Cape Cod, but many of the topics studied focus on questions being investigated as part of the larger research portfolio of scientists and faculty here at the MBL at sites ranging from the Arctic to the tropics and deserts to the deep sea. Roughly 1 in 5 SES students return to work with scientists in Woods Hole after the program as interns, field or lab assistants, as TAs in the SES. During today's webinar, you will hear from a recent alumna of the program, Lauren Wind, who attended from Allegheny College in 2013. As Lauren will explain, her experience comparing leaf litter decay and samples collected from Jerry Melillo's long-term soil warming plots inspired her to pursue a career in environmental science. Hungry for more research experiences, she pursued an NSF-funded internship at the Virginia Institute of Technology the following summer after completing the SES. And then after finishing Allegheny College, she was awarded a graduate fellowship to study biological systems engineering through the Global Change Center at Virginia Tech. And last week, she was awarded her doctorate based on her research on antibiotic resistance in microbial communities within agroecosystems. So now I'd like to introduce today's speakers. Unfortunately, Jerry Melillo, who had been scheduled to speak in this webinar, is today unable to participate. He's not feeling well. We wish him a speedy recovery. Jerry is a distinguished scientist at the MBL and founding director of the Semester in Environmental Science. He has been active both in research and policy arena, co-led 3 of the recent U.S. National Climate Assessments, and is a member of the National Academy of Sciences, past president of the Ecological Society, co-recipient of the 2007 Nobel Peace Prize awarded to the Intergovernmental Panel on Climate Change, and has many other honors too numerous to name. So in his place, Ed Rastetter will present some of the results from Jerry that Jerry was planning to discuss, as well as talk about his own research, establishing a framework for modeling the response of ecosystems to climate change. Ed is a senior scientist at the Ecosystem Center, is currently lead principal investigator at the Tuluc Lake Long-Term Ecological Research Program in the Arctic. And importantly, Ed teaches the mathematical modeling elective within the SES program. He uses simulation models to better focus research questions and design experimental work, as well as to predict the behavior of ecosystems. Ed will discuss how feedbacks between biogeochemical cycles and plant productivity could influence net carbon storage of ecosystems. Gus Shaver is a senior scientist emeritus at the Ecosystem Center, a plant ecologist and ISI highly cited researcher in ecology and the environment. Past board member and elected fellow of the Ecological Society of America, and past program director of the Division of Environmental Biology at the National Science Foundation, where he received the NSF Director's Award for Collaborative Integration. Gus will talk about his long-term field experiments in the Arctic. And now to begin, I'll turn the webinar over to Ed.
Thank you, Ken. Hello. There we go. Oops. So I want to acknowledge both the National Science Foundation and their program, the Long-Term Ecological Research Network, because most of the work we're going to be talking about today was supported by that means. So I want to start with a little outline of what Gus and I will be talking about today. I'm going to start off with a discussion of global warming and the global carbon budget. Then I'll present some results from Jerry Melillo on a soil warming experiment. Then I'll talk about some modeling constraints by nitrogen on the carbon sequestration in ecosystems. Then Gus will talk about the Arctic as a model system for testing some of the ideas about carbon-nitrogen interactions. And then Lauren Wind, Dr. Lauren Wind will give us a SES alum's perspective on things. So to put things into context, up at the upper left, you're looking at the size of the human population on Earth for over the last 12,000 years. And to give you an idea of just how fast it's growing recently, I've indicated what the human population was when I was born and what it is today. And just in my lifetime, it has tripled or almost tripled in size. The big graph is exactly the same data, but on a log scale so that you can see the patterns that at the low levels. And for a long time, down through here, the human population for the entire world was about the same as that of New York City today. And through time, there have been various technological advances that have allowed the human population to increase. And those technological advances have had ever expanding consequences for the environment. And the one that we're going to be talking about today is the consequences of the industrial age. So here is a graph of the CO2 concentration in the atmosphere over the last 10,000 years, derived from ice bubbles trapped in glacial ice in Antarctica and Greenland, and from direct measurements here in red from Mauna Loa. So the point is that the consequences of human activities now have a global scope. And the increase in the CO2 is a combination of both fossil fuel use, land use change, cement production and the growth of human population. So here are the major fluxes for the global carbon budget. Photosynthesis is around 120. That's balanced by respiration from plants and from the soil. And the oceans taking up and releasing about 90. And then there's a slight excess in the ocean, so about 2, and a slight excess in the terrestrial, about 3, that balances out land use change flux, net flux to the atmosphere of 1.6 billion metric tons per year, and the use of fossil fuels and the production of cement of about 9.9. Here are those data through time since the beginning of the industrial age. So the sources, here's fossil fuel and cement production. In tan here is the land use change, basically converting mostly forest into agriculture. And then down here are the sinks. So here is the ocean sink. In light green is the terrestrial sink. And a lot of it is showing up in the atmosphere. And the consequence of that, I want you to notice this, this sharp edge here, sort of slow and then accelerating at around 1950, 1960. And here is the temperature anomaly. That's just the temperature difference from the average from 1951 to 1980. And the temperature is growing it's warming at a rate of about 0.2 of a degree centigrade per decade. That means over the next century, it's going to increase about 2 degrees C if this keeps up. So one of the questions that we're asking is, can the terrestrial ecosystem store enough carbon to slow this accumulation of CO2 in the atmosphere and thereby slow global warming? So Jerry Melillo set up an experiment warming soils, and just to look at this idea just from the soil's perspective. And there were some unusual feedbacks. So here's his experimental setup. He set up 18 plots that were 6 meters by 6 meters in size. He set up 3 treatments in there. There was a control plots, which didn't have anything done to them. Then there were heated plots where he buried electric heating cables to warm the soil. And then there was what he calls a disturbance control, which is where he buried the heating cables, but he never put electricity to them. So they never heated the soil. And so he set it up so that the plots that were heated would be heated by about 5 degrees C. And here you can see in late winter, early spring, that the heated plots, these squares back here and the square in the foreground, the snow melts out earlier because it's 5 degrees warmer. So they're quite effective. And once the snow is gone, you can see that it's a little greener than the surrounding part of it, surrounding forest. So here are the data over the last 30 years or so. The controls are in black and the heated plots are in gray. So these are the flux of CO2 in grams carbon per meter squared per year out of the soil itself. And then down here are the difference. It's the heated plots minus the control plots. So this is the XX flux associated strictly with heating. And you can see that the experiment over the last 30 years went through 4 phases. In the first phase, there was quite a lot of carbon lost by the soil. Then there was a quiet phase, and then it built up again, and now it's going back into a quiet phase. And what we think is happening here is that in the first phase over here, the bacteria in the soil took advantage of the heating and used up all the labile carbon in the soil organic matter. And once that ran out, their metabolism slowed down. And then the community, the microbial community, restructured towards organisms that were able to use more recalcitrant forms of organic matter. And as their population increases, they start using that, and the flux comes out, and now they're coming back to a creation phase. So some conclusions from this is that warming does result in a release of CO2 from soils. The magnitude of that release changes over time. The magnitude decreases as labile forms of carbon are used up. The microbial communities are likely to change in response to the change in the quality of the organic matter. And this change in microbial community is likely to cause a second release of carbon. And we don't know about what happens after that. Now I want to talk about nitrogen constraints on carbon sequestration in terrestrial ecosystems. And I've put over here the major stocks of carbon in billion metric tons. So the atmosphere has about 750. Vegetation on land has about 560, but the soils have almost 3 times as much carbon and organic matter in the soils, and the oceans are a huge sink. Now we're going to talk about these two stocks here in the vegetation on land and in the soils. Now that's not evenly distributed across the globe, so I've put out 3 examples here. Now looking at a local scale, so the units are kilograms carbon per meter squared. So in a terrestrial rainforest, that might be 25 kilograms per meter squared in the vegetation and 18 in the soil. In a New England forest, like the one I just showed you that Jerry set up his experiment, there might be 11 in the vegetation and 17 in the soil. And Gus Shaver is going to tell you about Arctic tundra that might have about 0.6 kilograms per meter squared carbon in the vegetation, 12 in the active layers of the soil. But beneath that active layer in permafrost, there's a huge stock of organic matter with a lot of carbon in it. And that's one of the things we're concerned about. Okay, so the carbon that's stored in ecosystem is pretty tightly tied to nitrogen. You can't store carbon unless you have some nitrogen. So a typical forest in New England might have for every gram of carbon, every gram of nitrogen that you might have 120 grams of carbon. And for the soil, for every gram of nitrogen, you might have 25 grams of carbon in the soil organic matter. Now that nitrogen is constantly cycling around in the ecosystem. As the soil organic matter decomposes, it releases nitrogen. That nitrogen is taken up by the vegetation to support their production, the new leaves every year, any growth every year, and then it's recycled into the soil and litter, etc. And this cycle is very tight. The cycle itself might be 10 to 50 times as fast as any nitrogen coming in and going out from outside. So they're a major recycler. So how do you get around these nitrogen constraints on the amount of carbon stored in ecosystems? Well, the first and most obvious one is to just add more nitrogen. If you add more nitrogen, it gets entrained into this internal cycle, and you can store more carbon in the vegetation and in the soil. So you might fertilize, you might increase nitrogen fixation or favor nitrogen fixing species, or increase nitrogen deposition. The second way is to just increase the carbon to nitrogen ratio of the vegetation. So if you increase CO2 in the atmosphere, that automatically, when the vegetation responds to that, the carbon to nitrogen ratio of the vegetation tends to go up, but it's a small amount. The big changes come when you increase the amount of wood in the ecosystem. So that means getting bigger trees, allowing the trees to get bigger, or replacing herbs and grasses with trees. Taking farmland and putting it back into forest, for example, will sequester a lot of carbon. Now, obviously, the opposite also applies. So if you cut down a forest and replace it with a pasture, you're going to release all that carbon that's stored in wood back up into the atmosphere. The third way to increase carbon storage in the ecosystem is to increase the carbon per unit nitrogen in the soil, increase the C-N ratio of the soil, and just increasing the amount of litter. So if you increase the productivity of the ecosystem, that'll increase the amount of leaves that are dropped every year, the amount of roots that will die. Just increasing that will tend to increase the C-N ratio of the soil, because the C-N ratio of litter tends to be maybe twice that of the soil itself. So more and also higher C-N ratio litter. The final way around it is to move nitrogen around. So if you take 1 gram of nitrogen out of the soil by mineralizing it, and that you'll lose 25 grams of carbon. But if that nitrogen is then taken up by the vegetation, that 1 gram of nitrogen will be accompanied by 120 grams of carbon. So if you move nitrogen from the soil to the vegetation, there'll be a net increase in the carbon stored in the ecosystem. And one of the things that we expect of warming is that it will stimulate microbial activity, like we saw in the what Jerry showed in his experiment. So that releases nitrogen into the soil water that is available to plants. Also, the uptake by plants will tend to increase, but the litter fall is not as sensitive to temperature. So that tends to redistribute nitrogen from the soil to the vegetation. All right, so how do we deal with all this and try to make some predictions and hone our experiments, et cetera? Well, you can take what you learned in the field and what other people have published in the literature and try to describe those processes in terms of mathematical equations. And then take those mathematical equations, feed them into a computer, and let the computer predict where things will go. So that's what we mean by a computer model of an ecosystem. And we use maybe 4 or 5 different computer models here at the Ecosystem Center. I won't go into any details on any of them, but I'll show you some results. So here are those 3 ecosystems that I showed you early on in my part of this talk. And you're looking at the carbon gain in grams carbon per meter squared after 100 years of doubling of CO2, warming by 3.5 degrees, and increasing precipitation by 10%. And here are those 4 mechanisms: adding new nitrogen to the ecosystem, changing the C-N ratio of the vegetation, C-N ratio of the soil, moving nitrogen from the soil to the vegetation. Then there's an interactive term. So if you move nitrogen from soil to vegetation and you increase the C-N ratio of the vegetation, you get a little bit more. So in the tropical forest, the carbon gain is about evenly distributed between new nitrogen and this redistribution. Now, the new nitrogen is possible in this tropical forest because there are a lot of nitrogen fixing species in the vegetation. Here in New England, where we don't have that, most of the gain in carbon is from the redistribution of nitrogen from soils to vegetation. And in the tundra, where the vegetation is a very small component of the total organic matter in the system, most of it is in the soil, the increased production associated with elevated CO2 and warming gets transferred to the soil. And so you get an increase in the C-N ratio of the soil itself. So the conclusions from this section are the carbon sequestration in ecosystems is constrained by nutrients like nitrogen. Nitrogen is tightly cycled within ecosystems, and external sources are very slow relative to that internal cycle. So you can sequester carbon by increasing the amount of nitrogen, increasing the supply of nitrogen to ecosystems, increasing the C-N ratio by maybe making more and larger trees, increasing the C-N ratio of soil by producing more and higher C-N ratio litter, and by moving nitrogen from soil with low C-N ratio to vegetation with high C-N ratio. So now I'm going to turn it over to Gus Shaver, who's going to tell you about some experiments he's been running in the Arctic for a long time to test some of these ideas.
Thank you, Ed. Let's get set up here. Well, I'm going to talk about a series of experiments that we've been doing on the North Slope of Alaska for the last 40 years or so. It's been a great way to have a career in ecology, visiting Alaska every summer to study how carbon-nutrient interactions might limit changes in the Arctic carbon cycle in a warming climate. This picture is of our camp at Toolik Lake, Alaska. There's the lake itself, the camp. And the experiments I'm going to talk about were done here on the south shore of the lake, and over here, most of them, on the western shore of the lake. That's the Brooks Range in the background, and we're looking south in this image. So why is this important? Well, soils underlain by permafrost contain almost 1,700 petagrams of carbon, 17 times 10 to 100 times 10 to the 15th grams of carbon. That's about half of all soil carbon in the world. And it's more than twice the amount of carbon held in the atmosphere. Because of this, there's a tremendous potential for increased carbon release to the atmosphere, carbon accumulation in the atmosphere, if all of that carbon is decomposed as a result of global warming. Right now, that carbon is stable or relatively stable because it's cold. It's underlain by frozen ground. Much of the carbon is incorporated in the permanently frozen ground. But the world is warming, and the Arctic, in particular Alaska, is warming faster than any of the other regions of the Earth. And it's very important to know how this global climate change is going to impact positively or negatively on carbon content of the atmosphere. So what have we been doing at Toolik Lake? We've used a number of long-term experiments to show how temperature, light, and nutrient availability interact to control plant and soil carbon stocks. We have here a moist tussock tundra. You can see the tussocks of a sedge called Eriophyllum vaginatum here in the foreground. And then this is unfertilized tundra, unmanipulated tundra. Back here in the middle ground, we have a 5 by 20 meter plot of fertilized tundra, which has, it's a little bit taller, it's much denser, the canopy is, and it's composed almost entirely of Betula nana, dwarf birch, a deciduous shrub. Farther back, we have plots. This is just one of 4 blocks of treatments that we have in the field in this particular experiment. We have plots that have been covered by a greenhouse here and here. The uphill greenhouse is just that. It doesn't also receive any fertilizer addition. Downhill, we have a greenhouse plus fertilizer plot. These warm the air temperature during the summer by about 3 degrees centigrade, constantly. In the background, we have shade frames where we have reduced light intensity by using greenhouse shade cloth by about 50%. That's photosynthetically active radiation, that is. And the downhill from that, we have a shade frame that has been fertilized. What do these look like when you get up close? Here is the unfertilized greenhouse, same one shown in the main picture. And you can see here that after about 10 years of manipulation, the vegetation is certainly greener in there, but we know, because we measured it, that the vegetation does not have a higher biomass. In the background, though, we have a greenhouse in which we have combined the greenhouse treatment with a fertilizer addition. And we've had a tremendous increase in plant production. And of course, the same change in species composition, that is dwarf birch, in this greenhouse after 10 years. If we now look at what have we learned from these experiments, here are some results from the first experiment of its kind that we did, Terry Chapin and I did back in the late 1970s. We showed that plant biomass and carbon accumulation are basically a function of N and P accumulation in these plots. We have control plots here, unfertilized and fertilized. We have greenhouse covered plots here, unfertilized and fertilized. We have shade plots here, unfertilized and fertilized. I hope you can all see that my cursor here. This is biomass on the left, total nitrogen stocks in the middle, and total phosphorus stocks on the far right. What you can see from this experiment is that we only get a change in biomass when we add fertilizer. This is after 2 years of treatment. It doesn't matter whether the fertilization in this species is accompanied by a greenhouse or not. It doesn't matter whether this treatment was accompanied by fertilizer treatment was accompanied by shade or not. We only get an increase in biomass when we add fertilizer, and it's the same increase no matter what else the other treatment, the fertilizer treatment, was accompanied by. And we get no effect of greenhouse alone and shade alone. Those increases in biomass are roughly proportional to the increases in nitrogen stocks and the increases in phosphorus stocks of this individual species. So what happens when we let things go for a little longer time and we look at the whole community? This was done in our second major experiment, in which we began treating the plots in 1981, so that in 1983, we were able to harvest a control plot, control plot, a fertilizer plot, a warmed plot, a greenhouse warmed plot, a greenhouse plus fertilizer plot, and a low light plot after 3 years and after 9 years. The different segments of these bars show the abundance of different plant functional types. Non-vascular plants are open, unfilled segments. Evergreens are gray segments. Deciduous are these hatch segments. And grasses and sedges, graminoids, are in the black segments. What you can see here is, if you look at just the vascular components first, the mosses sort of have a mind of their own. They responded dramatically in the first couple of years, but gradually were eliminated from these plots over time. So let's just look at the vascular plants first. Fertilizer in this bar, the NT bar and the N bar, had the same effect, whether it was accompanied by a greenhouse or not. Warming had no significant effect on the productivity, above ground productivity of these plots. And light had no significant effect. This is 3 years or 9 years at half light. Light had no effect after 3 years, and it was just beginning to show a reduction in productivity at half light after 9 years. The other point to glean from this graph is that the effects of fertilizer in 1983 in both the greenhouse fertilized and the fertilizer only plot were exactly the same as in 1989, but the species composition, the functional type composition of those plots changed dramatically over those 6 years. And it also changed with respect to the control plots. We had dominance, as in this picture up here on the upper right, strong dominance by a single species, dwarf birch, Betula nana. So we had the same effect of fertilizer on these plots, no matter whether it was accompanied by a greenhouse or not. And it was the same effect in a wide range of mixes of plant forms. So changes in species or functional type composition have no effect on above ground primary production in these plots. Another important thing that we've learned from these plots is that the way the plant canopies are structured in these plots follows a consistent pattern. If we plot here the amount of nitrogen in leaves in the plant canopy against the leaf area of the plant canopy, we get a constant relationship. These open circles here are for control plots measured at 7 different times over 35 years in this long-term experiment. The closed circles here are from fertilized plots where we had big changes in species composition over 35 years. This highest dot on the upper right here is actually from the year 2000. After 20 years, 35 years, we came back down here. But the point is that the whole system is moving along this line, varying from year to year and with respect to treatment. There must be some very strong constraint on how these plants, how this vegetation has to build its canopy to optimize productivity. Going back to the 1990s, Ed Rastetter and Matt Williams found in their analysis of a group of plant communities, vegetations in the Kuparik River Basin near Toolik Lake, they found this exact same relationship and showed in a model developed by Matt Williams that productivity, gross primary production, increased as you go up the line. So there turned out to be very strong constraints on how plants' canopies are made in this system, and that all plants seem to be following the exact same rules in building their canopy. This is really quite important because it means that we can model Arctic systems using a single parameterization for all of the wide variety of ecosystem types that we have in the Arctic, dominated by evergreen or deciduous or graminoid species. Another lesson we've learned, a final lesson, is that after 20 years of nutrient fertilization, higher productivity is completely unrelated to the changes in soil carbon balance that we observed in these plots. Here you have from a paper written by Michelle Mack in 2004. This is productivity, above ground net primary production, measured by harvest methods in control plots in the open circles fertilized plots in the filled squares, productivity in these plots has been doubled for 20 years. And presumably the litter resulting from that productivity has been falling on these plots for just as long, and leading to soil formation, soil organic matter formation. But what we find here is that we've actually lost carbon from fertilized plots over this time. So primary production is doubled, but we've lost about a third of our soil carbon from deep in the soil. We've lost about a third of our soil carbon from lower portions of the organic mat. And we see the effect of litter accumulation only on in the upper few centimeters of soil. So higher productivity does not lead to greater carbon accumulation in tundra soils. We did this again in 15 years later, in 2015, and found that we're now starting to accumulate carbon again in soil. So we have a lot of work to do to explain what the relationship is between primary production and plant and soil carbon pools in these tundra systems. A final point is that these experiments have taught us a whole lot about how carbon and nitrogen cycles interact with each other in tundra systems. But we're also starting to see, as the climate changes, wildfires. You may have heard about huge wildfires in Siberia last summer. We're seeing these wildfires on the North Slope. And we're also seeing that as the climate warms, ice in soil, the permafrost, is thawing. And we have a wide range of mass wasting and landslip processes that are providing patches of intense carbon loss in these systems. And in the long run, it may be these large-scale disturbances that actually contribute more in terms of carbon inputs to the atmosphere than the direct effects of climate change on otherwise undisturbed systems. So how do carbon-nutrient interactions limit changes in the Arctic carbon cycle in a warming climate? Plant biomass and carbon accumulation are tightly linked to NNP accumulation, irrespective of temperature or light. There is no species effect on total biomass or carbon and nutrient content of whole vegetation. Canopy and canopy leaf area in relation to canopy nitrogen content is constant. Essentially, all canopies function in the same way, which makes modeling of pan-Arctic carbon balance much simpler. I didn't show you that warming alone restructures these soil communities and carbon distribution, but it has no net effect on total carbon balance. Increased productivity of fertilized plots does not lead to higher soil carbon stocks. Disturbances like fire and thermoscars may in the long run have even greater an impact on carbon stocks than changes in climate or nutrient inputs. Like to leave this with a picture of just a few of the students who have helped us with this work over the last 40 years. Several of these are SES students. We have that, there. 3 SES students from 1 SES class, one there, one there, one there. We have a lot of fun, and we owe these people a great deal for their help and enthusiasm. Now I'm going to turn it over to Lauren Wind, who's going to tell you about her experience with SES.
You're muted, Lauren.
You'd think by the end of 2020, I would have figured that one out by now. But thank you for having me. Like Gus said, my name is Lauren Wind, and I was in SES in 2013. And I was really lucky. I got to work with Jerry Melillo, and I worked up at those Harvard forest plots that Ed had talked about earlier on in this talk today. And I looked at the decomposition rates of leaf litter from the leaves that have fallen from the trees that were grown in the heated plots versus the control plots. And what I really am glad that I was a part of SES is this was my first time to design my own experiment. And that's really what jump-started the career and where I am today. And what I did was I was able to measure the amounts of nitrogen and carbon in the leaf litter. And how we did this is we tried to speed up decay rates of leaf litter. Now, SES is a semester. And during our time of our experiment, we only had 3 weeks to look at the decay rate. So how we sped it up is we made this leaf tea, and we input these bags of leaf litter based on the different types of tree species and leaf species into these hot water baths to try to increase decay rates. And what I was able to find in our results here is that we see different decay rates based on how much labile or recalcitrant carbon are in the leaf species and the tree species. But 23 days, even when we made our leaf tea, was not enough time to see decomp. But I think that the main part of this was just me getting my hands on it and doing my own experiment. It took a lot of work. And what I really liked about SES when I was there is that we were really collaborative. I couldn't go to the Harvard forest by myself, so I brought a few of my cohort students with me as well. I had a lot of help in the lab to sort all the leaf species. And SES is just a really fun time. These are some of my favorite photos from the time that I was there. In the top right-hand corner is when Ken invites us all to his house for a Halloween party. In the bottom right-hand corner was my favorite field trip when we went to Plum Island and were able to kayak in the estuaries. In the middle was when we took Joe's class and we're making our microcosms, and we had to collect our samples and our sediment there. We ran the relay at the Falmouth Relay, so the Cape Cod Relay. And it was my first time touching a blue crab. I am from Canton, Ohio, so I didn't have that experience before, and that was really great. And then, like I said, that was my first time to do my own research, and that really got me jazzed to go to grad school. And so I went to Virginia Tech, and I studied biological systems engineering. I got my master's in 2017, and like they just said, I successfully defended my PhD last week. So I'm really excited for what's to come next. And I studied antibiotic resistance in our agroecosystems, and I did this at the field scale. And one of the things I thought that was really amazing about working at the Harvard Forest, and also just hearing about Gus's research and Ed's research up in the Arctic and all the other projects that are going on at the Ecosystem Center, is these long-term real-world experiments matter. And it wasn't even until after they were going to stop the experiment and Jerry's experiment up there that they saw, oh, maybe we should keep doing this for a few more years and we'll see different phases. And they didn't know that until they kept doing it real world, long term. And I think that just speaks a lot for studying climate change in the future, that we need to keep looking at trends, because we are seeing very interesting things. And so that's my experience with SES and where I am today. And if anyone has more questions about joining SES, you can go ahead and visit the links here or email Ken. And I know that Linda said that today's the day to sign up. So that's very exciting. Thank you.
Thanks, Lauren. So we are now opening this up for questions. If anyone out there has a question for either Lauren, Ed, or Gus. The first question I have is for Gus, and it asks, have you measured soil respiration in the greenhouses versus control sites? If warming itself doesn't stimulate carbon storage in the vegetation, but just stimulates soil respiration, wouldn't that suggest that you're going to accelerate climate change by releasing more CO2?
We've done a lot of work on soil respiration, including a number of incubation studies that, with Newt Nadelhofer and Ann Giblin. We what happens is that respiration is increased by fertilization. I didn't say it, but one of the really interesting things about that fertilizer experiment is that in the long run, we lost both nitrogen and carbon from our long-term fertilizer plots. We added over 20 years, the first 20 years, we added almost as much nitrogen to those plots as there is in the ground in the control plots above the permafrost. And we ended up with less nitrogen in the soil profile in those fertilizer plots. So we lost more nitrogen than we had added over 20 years. We had an SES student, Marcel Alexander Ozinskas, who came up and did a master's degree on that. And we think part of that loss of nitrogen may be because we denitrification in these plots. We were adding ammonium nitrate to the plots for a long time. And we also know that we primed the carbon decomposition, organic matter decomposition, by adding nitrogen. Josh Schimmel thinks these decomposition in these systems is nitrogen limited as well. So yeah, there were a lot of effects on soil respiration.
Okay. Thanks, Gus. So for Ed, the question is, did I understand correctly? You are predicting an increase in carbon stocks in the tropics of 6 kilograms per meter squared in 100 years? If so, it's critical to preserve these terrestrial or these tropical forests, tropical systems.
Well, that's certainly what the model is saying right now. The problem, of course, is do we have enough data that is sufficient to constrain the model, and are we driving it with the right drivers, right? So that modeling experiment was done with a standardized increase of 3.5 degrees C warming. We know that the Arctic is warming pretty fast. That might be a little bit too fast for the tropics, but we wanted to be able to compare the tropics to the temperate to the Arctic without based on the structure of the ecosystem as opposed to the climate change used to drive it. So what the model says and what will actually happen could be very different. I'm not.
So follow on that is are there, based on your modeling, any practical or management approaches that you would recommend for enhancing soil carbon storage?
Yeah, so there are a lot of people that are working in agricultural systems to try to do that. Try to, low-till agriculture, so you're not aerating the soil and stimulating decomposition, so you keep the organic matter there. Certainly, reforestation is a way of storing carbon over long periods of time. I know that Jim Galloway is on here, and so adding nitrogen to an ecosystem will store carbon, but only if you can keep that nitrogen in the ecosystem. If it goes in and then just simply dribbles back out, then it's going to run into aquatic systems where you don't get anywhere near the carbon gain associated with it.
And a final question for Lauren, which is, what's next now that you finished your PhD?
Well, I am fielding some postdoc opportunities internationally or domestic. So I just need to figure out what I'm going to do after the holidays and still be in research doing antibiotic resistance. That's the plan. Thanks, Ken.
Okay. Well, it is 12:55, and we know that most of the folks watching, plan to spend about an hour on this. So I think it's time to bring things to an end. But I want to say that we've thoroughly enjoyed having the strong participation by all of you out there in webinar land. And I'll echo Linda's comments that this is a banner day for education, as we are now going to an online application process, and SES is the first of the MBL courses to have its application go live. So thanks everyone for both participating in the webinars and for watching. And we especially appreciate the fact that so many of our alums were willing to come back and talk in the webinar series. You guys are what makes SES really fun for us as faculty and rewarding. So thanks to all of you. Bye.